to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 58th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I have been in the bioanalytical LCMS space for over 27 years. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm the Senior Scientific Advisor for Ligand Binding Assays and Biomarkers. I've been here at KCS for 9 years, been in the industry for upwards of 28 years now.
Dom and I are Senior Scientific Advisors at KCS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCS's services. I'm located in upstate New York. Dom is in Kansas and our producer is Missouri, so we're all over the map. We are thrilled to have you listening to the 58th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 58, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, John, we're going to continue with what we had started in our last podcast. So we're going to do part 2 of the evolution of drug design and its impact on bioanalysis. Of course, we'll always start with news and resources as we've done since inception. We'll go over the latest news on COVID, and then we'll go over some of our favorite articles on non-COVID news, and then we'll talk a little bit about investments and some really near and dear stuff around what people are doing in the rare disease space. So, John, with that, let's just jump right into it.
So, Dominic, how about starting with the news?
Yeah, John, as always, we'll talk about COVID, and the first item will be the national COVID admissions tick upwards. So this isn't surprising, John, right? So there's U.S. hospitals have seen a steady uptick. I don't know how concerning it all is, but I think COVID's here to stay. But, you know, from certain second week of July or through mid-July, there's been like almost a 15% increase, almost up to 20%. I think, John, this is part of what we would expect. I think natural immunity is kind of kicking in. The deaths are certainly down, but you know, I'm cautiously optimistic. I'm okay that, you know, I think everybody should be comfortable that it's kind of going to linger, but I guess what I don't want to see is shutdowns, John, but you know, maybe you can share a little bit more about it, but I don't think this is surprising to see some upticks because we're just doing more too, I think.
Yeah, I don't think we're going to see shutdowns, but I am a little bit alarmed that we're so complacent about 400 deaths a day or 350 deaths a day as we're reporting right now. I mean, yes, obviously it's here to stay, but it is disrupting life. You know, it's like I'm still regularly seeing messages from friends who've got it for the first time. We talked last time about, you know, putting on the concert here last Friday, and I had a few people paid ahead, and I got a message from one woman who said 2 days beforehand, yeah, I paid but can't make it because I got COVID. So it is sort of disrupting lives, etc. I think the message here is really, it's sort of just hammering what we've done all the way through the podcast, is if you haven't got vaccinated, get vaccinated. If you're due for a booster, get a booster, because the variants is obviously still evolving, and the dominant variant is BA5, that there's concerns about how effective the vaccines are, but at least it's going to be some protection. And you know, there's obviously the drug or the vaccine manufacturers are working on updated vaccines to try and address these later variants. But the more we can do to get people vaccinated, the more we can suppress this on the whole, and hopefully these death rates go down, because I don't like seeing 350 people, you know, dying.
You know what, John, I don't want to.
Not a day, so it's not as bad.
It's in a week. Yeah, it was just a spike that week. You're right. I don't want to take away from that number. It is slightly alarming, but I do know that this isn't, you know, unfortunately, senescence is part of life.
Sure.
With the heat and all that's happening, I don't necessarily want to just be skeptical and question the number, but it's not uncommon this time of year to have quite a few passings due to even like summer colds and stuff like that, because it's harsh time of I
sure.
think of it as a harsh time of the year. So, but I do agree with you that we shouldn't just forget about it.
Yeah.
I hope in general, I would really like to continue to see people practice all the things I was practicing long before COVID got here, John. Like I was always washing, not always, I wasn't like walking around with hand sanitizer, but I routinely washed my hands. I didn't want to touch my eyes. I was always on my son about not putting his hands in his eyes or his mouth unless they were clean, and don't touch doorknobs, just subtle things. I hope we are keeping up that level of cleanliness that we had seen at its peak. And I wonder if that's part of it too. Remember at one point it was like you'd walk into a place and someone would be scrubbing the doors or clean. Like, I don't know if that's going on as much. I hope we can get back to it, but it is something that, we're going to monitor it. And I'm with you. Get vaccinated. I don't know if you need all 4 shots, but at least get a couple. And then, we'll probably talk about this at the next podcast, but I did read about a nice upcoming vaccine. They're really getting ahead of it in terms of trying to have next year's one. We got a lot of in silico science. We got a lot of advancements in this space. The next round of vaccines, not only are they hopefully going to be protective, maybe a little bit more than what we had now, and potentially even looking towards evolving with it. That's some of the technology we have.
And certainly looking at different drug delivery approaches. So if you don't like needles, they're trying to work on oral vaccines.
And there's like a virus-like particle coming out that has promise. There was a whole gamut of vaccines that were out there outside of just Pfizer's and Moderna's that are still advancing the ways. There's some real good virus-like particles. That's the Gardasil type vaccine for HPV. All stuff that, just the comfort level is what we want. There's still a lot to be learned, but if you have alternatives, I think you should do something though, because maybe it doesn't protect you 100%, but there is still good science that says it helps with, if you do get infected, giving it a milder case. But it's something we want to keep an eye on.
I think the reason for including this was, don't forget it's out there. Just do what you can to protect yourself. I agree on the hygiene piece and everything else. You can vaccinate, do that. And it's just be aware of it, because it hasn't gone away. It's like it's sort of died down in the media, but don't ignore the fact that it is still there, and people are still regularly getting infected.
It's good to keep a couple feet from each other. I was always a big proponent of that too. I was constantly just like, I remember throughout the 90s looking at people when we were out at a bar or something being like, do you know how filthy it is in here? And people just being like, man, you got to just shut up. But I'm like, no, you don't understand. So,
Get out of my face. Yes. Let's move on to the next one.
You bet.
Well, this is Pfizer considers antiviral options as Paxlovid use skyrockets and amid worries of resistance. And this is, it's Paxlovid. Obviously, it's a oral drug, so it's a convenient thing to administer when people are infected, because it obviously helps reduce the risk of hospitalization. So it's certainly got its uses. But as with any antibiotic or antiviral, there's a concern of the build-up of resistance. So I think this article is saying, well, to begin with, they were fairly slow implementing Paxlovid. Now it's like there's a real push to prescribe it, but there's actually a number of groups that really it shouldn't be prescribed to, as both my wife and I found out when we sort of asked about it when we were infected with COVID. The hospital were very good and walked us through a screening and said, we don't recommend that you take that. So it was sort of really, because I think according to the articles, 23 counter-indicated, 35 different drugs are contraindicated with Paxlovid according to the FDA fact sheet. So there's a number of interactions to be aware of that maybe you don't want to be taking it if you're taking any one of those 5 drugs. So it's interesting that, it's like as you solve one problem, then you have you solve one problem, but then downstream there's another problem that you have to solve, and so it becomes like, well, we've got Paxlovid as a first line of defense taking this approach, but we also have to be watching what's the effect of us prescribing there, and then what else is in the pipeline that could possibly circumvent any resistance to Paxlovid if that occurs. I mean, there is obviously debate about the resistance piece. So some virologists are concerned about it. I think it was a Pfizer spokesman said, it's like, well, it's a fairly restricted pathway, so it's challenging because of the mechanism of the drug for the virus to mutate against it. So I will step back and let you have
Yeah, no, you said a lot of things in there. Paxlovid is, the president took it, Biden took it. That made, they say here in the article, that's like 40% of all prescriptions have been taken in June and July.
Sure,
I think that was part of it, which is good. It's really interesting. The COMED, that's how I think of it, right? That's what this is. There's some, if you take it along with other medications, and that's definitely something that know your health, John. I think that's a hard one. If you're on medications, be very, make sure you don't hide it or point it out to the doctor. And
exactly.
And so
be upfront about those.
It sounds like you had really good care in that. Your doc was kind of like, hey, look, this could be, so that's wonderful. But obviously, if you're recommended to take it. And then the idea of resistance. This is there's certainly antibiotic resistance. We know that. Viral resistance is a little cloudier, although I think for me, there's, I kind of come at it from a little bit of an evolutionary, like philosophical, that's kind of how I do like facts and numbers, but some of it does require some, real pensive thought.
Yep.
And to me, there's so much redundancy. There's so much, the body's so strong, and, these pathways are so narrow that there probably will be resistance. That's what I'm trying to say.
Sure.
I do think the body, if you take something routinely, starts to diminish the effect of it, just like taking, say, creatine, for instance, or
I don't know if you've seen that, but I saw that with like Claritin, and then in terms of those anti
it's all receptors, right, John?
And so with seasonal allergies, I started with Claritin. It was great to begin with, and then it got to the stage where it was having no effect at all. Then moved to Zyrtec, same cycle, and again it sort of after a while stopped, and then Montelukast is now my seasonal.
Yeah, maybe go back to Claritin though, right? You might be able to go back to Claritin after this. So that's exact. I think you're even given some real life examples of stuff, and I mentioned creatine because I think that has the same effect. I don't take a lot of meds, John, but I know, like, when I was, kind of into that, after a few months, it just didn't work as well, right? You kind of got this. So it's all receptor-based stuff. That doesn't change how, even though this is more passive, right? These aren't, I say receptors, I mean, small molecules can make their way kind of passively into a cell, but they still got to bind to something and cause some mechanism action that over time you're going to diminish. If you keep hitting that, the body starts to downregulate it. And so that's, I think it's inevitable, but I think the good news is we've got other sorts of antivirals that could be used, but if someone's telling you to take it, I say take it. But
sure, I agree. I just think it's interesting. It's like it's certainly. I think the message from this is that like you said earlier, be open about the drugs you're on. Your doctor should have those records anyway. But if it comes to discussion, well, and when it came to us, the conversation was with the third party, and actually, so it wasn't our standard doctor.
Got it.
Yeah. So, but they were talking about what are you on, and then atorvastatin was the one for me that's like, you shouldn't take if you're taking atorvastatin. I was like, wow, I'll drop atorvastatin for a while. But anyway,
no, but I think every once in a while I'll take some Prilosec or omeprazole, and so that's not always prescribed. And sometimes last time I went to the doc, they asked for my health check, and I said I wasn't taking anything, and then I remembered 10 minutes into it, I go, well, wait a minute, I guess I do.
Sure.
Omeprazole, I'm funny, John. If I know I'm going to go out maybe tonight and have some cocktails, I'll pop one in because that's it only upsets me when maybe that's too much information, but the only time I need it is if I feel like I'm going to be drinking, which is any day that ends in Y, John. No, all kidding aside, this is good stuff, John. I think we can move on.
Let's move on. Absolutely, yeah. I just thought it's all I think COVID is relatively old news, but it's always interesting to see just the little snippets that pop out around.
Go ahead. No, go ahead. That's good. We'll talk about some non-COVID stuff, John.
So the next non-COVID is dissolving nerve cooling device offers drug-free pain relief. And this, I like this because obviously if any of you have chronic pain conditions, often you're going to be prescribed opioid based drugs that carry their own risks, obviously, of long-term addiction and things like that. And so there's a group in Northwestern University have looked at finding actually a mechanical way of addressing this. So they published a study in Science recently that describing a new device that is implanted near the nerves to cool them down, blocking pain signals from reaching the brain. So the principle is it's like the implant goes directly into the bod. It's completely absorbed once it's no longer needed. But it's a cuff on one end wraps around a single nerve at a time. Once in place, separate channels of liquid coolant and dry nitrogen flow into a shared chamber, immediately causing the coolant to begin evaporating. The cooling process of evaporation lowers the temperature of the nerve, numbing it enough to stop the transmission of pain signals to the brain. So I think, to me, this is like a really clever little piece of engineering, and so it's basically, they're the way they approached this was where they were looking at exploiting mechanisms that have some similarities that cause your fingers to feel numb when cold.
Yeah,
And
John.
I'll pass it back to you.
No, I think this is. I love this stuff, right? Not only as I start to age a little bit, and I'm experiencing for the first time in my life any sort of, I got a little bit of a back issue, but this comes from, I don't know how much you know about a couple things, some advancements the NFL had. So there was spinal cord injuries in the NFL. Obviously, they've really dropped that down in terms of equipment and hitting and all that. But when they did happen, they made some advancements in that on-field procedures when someone had a hit that they worried about some sort of severance of the spinal cord or any sort of thing that may have made someone paralyzed. They started infusing liquid nitrogen like this is essentially liquid nitrogen.
Yeah, absolutely. Yeah.
Started doing that, and that allowed for a handful of players that normally would have been either paraplegics or completely paralyzed to allow it. Now they're not playing in the NFL, but they're living a normal life because of this type of cold treatment to abate the inflammation. That's what this is doing, right?
Yeah, absolutely.
So
yeah.
It's stemming from that. And then, of course, I don't know how much you know about cryopreservation treatments, where these are where I have a gift card. There's a couple of them here in Kansas City. You go in and you put in pants or a jacket, and they profuse liquid vapor into those pants, and it's to help you heal. So this is stuff that's kind of, you can proactively go get it. The fact that they can insert this sheet of paper around the nerves, I'm like, dude, John, this is like bionic man stuff, right? Or bionic person stuff. This is fantastic.
Yeah, absolutely. Yeah, I mean, to me,
the one thing I can't quite tell is they say they can control it, but is it a one-time thing and then it's there permanently, or do they have to re-get it? I can't quite tell. But
I'll tell you that. Well, basically, the temperature drop is monitored by a tiny sensor embedded in the device, so it transmits information so that the patients and the healthcare can control the therapy based on the feedback from that sensor. So ensure the temperature isn't dropped too much, or so if it goes too far, then it could damage tissue and things
of course.
like that.
Yeah, like anything. But so this is a one-time sort of surgery. Okay, and then it's like there's enough in there that they can control it. I mean, you don't need enough, so it doesn't. Do they have to replenish it? But let's not worry about
that.
No,
well, I suppose each piece is a one-time administration, because once the treatment period is complete, the implants completely water soluble
got it.
are fully and safely absorbed by the body's biofluids over the span of days and weeks. Like, to me, like I always love looking for these things. What is our potential future? And this is, to me, this is fantastic.
Right now I want it on my ankle, my lower back, my elbow.
Sure. When you talk about back pain, and the number of times, most of the time I'm fine, but you can do the tiniest thing and trigger your back, and it can incapacitate you for weeks at a stretch. So if you get something like this, that would be fantastic. And to me, it's like, you're right. It's future stuff, but to have.
But it's here now. It's like this is like almost, I don't know, not Matrix like, but it is like the fact that we're inserting stuff into people. I think a lot of it comes from the diabetic pumps too. We've made great advancements from those types of things to allow for this type of technology. But John, it's funny you say it can be debilitating. Well, the problem with me is I refuse to let me rest at all. So even though my back hurts a little bit, I'm still swimming and running and doing things. And like someone's like, why don't you go see somebody? And I'm like, the first thing someone's going to tell me is to rest. And you think I would do that, but that's neither here nor there, John. But this is a great story. This is why we got into science. It's part of the. I hope our audience appreciates that. Go find out what's out there. That's part of why we're on this podcast right now. In addition to helping out bioanalytically, but I hope everybody appreciates that there's some great stuff. And go talk to your docs and educate your doc if you need to on some of the things that are out there and available. So with that, John, I'll get off my I'll get off my soapbox and we can move on to the next.
One thing it doesn't make clear is if they have a trials coming up to really advance this. I think it's just talking about technology, so I don't know what the next.
Well, the device world's a little different, isn't it?
Yeah.
In terms of they still do trials, but you're right. But it definitely has a different path than say a small or large molecule.
Yeah. So anyway, I like to say, let's move on to the next one. And this is Nestle puts down $40 million, kicks off deal with gut microbiome specialist to fight food allergies. It's basically French company Enterome, which works with the microbiome, has received $40 million in a wide-ranging deal with its longtime Swiss investor Nestle Health Science to co-develop an oral pill for food allergies that's on the cusp of entering the clinic. So this again is a new piece, and Enterome's background is they basically looked at the microbiome. So basically looking at human gut bacteria, looking through all the different proteins.
That's a dirty job, John. No, sorry.
Again, sorry. That's a dirty job. Absolutely.
Yeah, that's a dirty job looking at all that fecal matter. Sorry, I didn't mean to.
Yeah, so basically they went through it, and they screened thousands of molecules there to try and find a molecule for. In this case, they were looking for something that mimics interleukin-10 because interleukin-10 is known to fight food allergies and conditions such as inflammatory bowel disease. So they found a small protein. They dubbed it EB1010. It's going into clinical trials. And so it says they scanned the gut microbiome of 26,000 people worldwide using their fecal matter. I'd love how the language cleans up really what they were doing. It's like I don't even want to think about that. But they found 23 million unique proteins and peptides manufactured by the gut bacteria. And actually, again, I know there are other companies out there looking at the microbiome, but this is, again, look at the potential there for possibly other compounds that, for dealing with other conditions that are there. This is all fresh and new stuff. Well, it's not fresh because it's your gut, but it's all new stuff.
No, it's good. Well, it's funny because TNF alpha was the hallmark for inflammatory bowel disease, and there are lots of available drugs to help mop that up. Although one of the major challenges of the gut is it's a different immune system. It's kind of got a different. It's not the same as circulation. It's got a much harsher environment. There's all sorts of technology out there to help keep TNF alpha stuck to like goblet cells within the stomach so that it can help treat or mop up, or anti-TNF alpha, sorry, the opposite. And this is slightly different in that they're using an IL-10 mimic, but they instead of trying to mop up the pro-inflammatory cytokine TNF alpha, and people would take, that like Enbrel is an anti-TNF alpha protein, and they would add motifs to it to get it to stick to cells of the gut. Real cool science.
Sure.
Well, this is even better, right? They found interleukin-10 and IL-10 is it's like a T0 cytokine. It really falls into that transition to stop. It is known to stop the inflammatory response, but too much IL-10 can be bad too, because that can lead to chronic. But in this case, they found this 30 amino acid protein that mimics IL-10, and really the power of this is it's like you said, they talked about 26,000 people. They looked at 23 million unique proteins, John, in order to manufacture the one they had. I'd take this drug. I bet you it works really well. Yeah. Really cool stuff.
So they basically say they're gonna. I mean, they're looking for it to be orally dosed.
Yeah.
Enter clinical trials 2023 with the hope that they can have something to market in 2028. So I mean, yeah,
My only advice to him is try to maybe add something to it that gets it to stick. But I probably anyway. This is great stuff. They'll continue to evolve this.
yeah,
Great.
they're looking to they're looking to work further in this partnership.
Yeah, you got to keep the half life
Other-
longer, right? It's probably got a short half life.
Yeah,
You wouldn't want to pegylate it, but like I said, there are motifs that are known to bind to gut cells, so that it would, it's got to be in the site of, it has to stay in the microbiome to be
sure.
effective, right?
Yeah,
And that's
yeah.
like any drug. We've talked about that in the past. That's what makes ADC so powerful, right? If the antibody can bind to the tumor and bring the small molecule with it, that becomes much more of a potent drug. Same thing with these types of drugs, but they're on their way. This French company, Enterome, hats off to you for, blood, sweat, and tears looking at fecal matter. And John, I've done a few of those, and they're not my favorite things in the world.
No. I mean,
Vicks
yeah.
Vapor Rub goes a long way above the nose. If you know what Vic Vapor Rub is, that helps you.
So the longer term is they hope to apply this kind of approach to address things like allergens, just like peanut allergies and things like that. So
I went towards the gut, but this is an allergic response.
Yeah,
Sorry, but I still think
it's a longer term plan. But yeah, I like to say I will watch this with interest. So shall we move on to the next one? Again, we're talking sort of really groundbreaking stuff, and this is gene editing for heart disease. Verve kicks off landmate. Oh, I can't even speak now. So excited. Gene editing for heart disease. Verve kicks off landmark base editing trial. So now we're back into the cell and gene therapy arena. And this is Verve Therapeutics has dosed the first patient with its lead cardiovascular drug, marking a major milestone for next-gen gene editing technique known as base editing. So they've started a clinical trial in New Zealand, and this drug is designed to permanently turn off the PCSK9 gene in patients with heterozygous familial hypercholesterolemia who have dangerously high cholesterol levels. And again, this is, we talked about, all the different approaches for gene editing, but this is a new approach. They've in-licensed technology from Beam Therapeutics, and you want to talk through the base editing pieces? You're closer to the stuff.
Yeah, no, I mean this is really cool stuff. It is a little hard to kind of wrap my head around it without talking to David Liu or somebody there to really get, because they don't give you this is some proprietary stuff.
Sure,
right?
Absolutely.
Yeah, I think just what you said there, rather than trying to do what CRISPR is doing, they're taking a different approach. Instead of cutting it, they're going to look at chemically changing it, and it doesn't quite
a single base in the
yeah.
I mean,
they don't go into how, John, right? But there's a lot of tools that allow, like, for instance, you got a lot of natural proteins in your body that are gene editing tools that they'll kind of give a QC after DNA is made, and if they see an A is put where a T is supposed to be, they'll flip it. Yeah. So I imagine that's part of what's happening here. They're probably capitalizing on some of those proteins that do that. I don't know how much our audience or even that, like, even as we speak, there's these, your body's dividing and there are these little proteins in there QCing how the DNA is going, and they'll pop out the wrong one and put in the right one. So to do that at like a strategic level is, that's like Star Trek stuff to me. That's unbelievable.
Yeah. So basically, the first in-human trial, dubbed Heart-1, will enroll around 40 adult patients with established atherosclerotic cardiovascular disease. There'll be a single ascending dose followed by expansion cohort receiving the optimized dose and an optional second dose group for those who had received a lower dose in the first part of the study. So, the readouts next year, and we'll see what happens beyond that.
No, good stuff, John. That's again, this is wonderful science that we're covering here. I hope everybody appreciates that.
So let's move into rare disease stuff, and this is it was on Endpoints, but it's actually a Kaiser Health News article about parents become drug developers to save their children's lives, and this is really talk about the challenge of rare disease medications, and if in many cases that parents have a child who who isn't developing it as normal, and when it's diagnosed for some, in some cases, it turns out to be a disease where it's a very, very small population, and the challenge there is there's so many rare diseases out there. The pharmaceutical industry has only really scratched the surface of rare disease, and also as commercial businesses, it's that challenge in terms of developing a drug for a tiny population. How do you price it if it even gets to commercialization? And then will then the health authorities be willing to pay that once you've priced it a certain way? So there's that investment / return challenge for pharmaceutical companies. So some parents have gone the route of fundraising to address the rare disease that affects their child. It cites this one family called the Carmichaels, who their daughter was diagnosed with PMM2 CDG that affects fewer than 1,000 people worldwide. Her parents raised $250,000 for scientists to screen existing drugs to find a potential treatment. So this wasn't going back to basics, but looking, is there something out there that could be repurposed to address this? And they found something that showed promising results. They then set up a foundation. The Mayo Clinic, called Maggie's Pearl Mayo Clinic, are involved as well, and they're trying to advance that. But that's just one condition, and there's a number of other examples in this where other people have taken different approaches. But I think it's a very interesting part of the drug development spectrum. And
there's so many things. This is a very good article, John.
It really is,
it's very informative. The fact that only, there's some good stats just on clinical trials in general. Only about 12% of drugs in clinical trials are ever approved. So think about that. That means you're failing roughly 88% of the time.
Yes.
That in and of itself, I hope people understand how challenging it is to get a drug approval. Going through all, preclinical to phase 3 is a long endeavor for a 12% hit rate, John, right?
Yeah. So even if they've repurposed the drug and it shows initial promise, there's no guarantee that initial promise will translate into long-term efficacy and approval. So I mean, there's so much. Yeah, it's like you said. Yeah, there's that.
And then of the 12% of the drugs that are approved, only 12% of those are focused on rare diseases. So it's really, this is still an unmet need. It says in the article, it says something like 95% of all rare diseases are without an FDA approval, yet 1 in 10 Americans are affected by this. So it's like, wow, it's really rare, that 1 in 10. That doesn't seem rare to me, although there are 7,000 rare diseases is what this article cites. So all of that, these are the companies that do this are fabulous to work with, John.
Oh, agreed. Absolutely.
This seems to be, and I'll let you take it back the mic here, but this article talks about it's not for profit, and then they did something very creative to work around that, didn't they? In terms of creating a cryptocurrency, right? That's
another group that So the Carmichaels is one piece. We can touch on the.
Yeah, no, I think we should, because I see that there's two separate things.
Oh, there's actually like 3 different sections in this. I think it's just to give a little bit more on the I'm sorry, I've lost the name again. Maggie's Cure is Maggie's Cure, or Maggie's Pearl. Sorry, just give a little bit more on them. They basically are moving forward to the Carmichael family's help to pay for clinical trial estimates will cost $3 to $5 million. But they are getting help from the federal Small Business Innovation Research Grant. So there is, but they've got to fundraise somewhere. It's not all going to come from that one grant.
Yeah, I see.
So that is moving forward, and like you say, their focus isn't on making profits. They're actually running this more as a business, although it's a foundation. They're saying we're not a traditional biotech with shareholders, but that have certain profit thresholds.
Well, you have to, John.
They're not focused on profit. However, they have pledged to reinvest a portion of its profits into research and development. The rest would flow to the ventures owners, including the Carmichael family. So obviously, their life has been upended, and they're taking the direction of helping to find a cure. And
John, even if you're for a nonprofit, you got to function like a biotech in order to compete.
absolutely.
You can't compete in this market without being that like running like a biotech. But so, John, maybe we're getting a little long here, but this is some. I do think we should touch on what Vibe Biotech is doing. And
Oh, sure, absolutely. So, yeah, Vibe Biotechnology are based in Boston, announced a cryptocurrency-based model to raise money for rare disease development. Investors will have the power to vote on rare disease research proposals, and patients' families have ownership stakes in promising therapies. So that's basically combining a lot of Carmichaels into one pot. So
well, that's funding, right? I think that's
exactly.
really cool, right?
But then the choice is made, but I assume it's by vote. And so they have actually launched two biotech companies with two foundations: Chelsea's Hope, which focuses on Lafora disease, a fatal form of progressive myoclonus epilepsy, and NF2 Biosolutions, which hopes to accelerate a gene therapy for neurofibromatosis type 2, which causes the growth of non-cancerous tumors in the nervous system. But they also talk about part of the issue right now is we've seen this downturn in the biotech industry. So another example is companies and families passing on the technology to companies, but this has happened with the Cure Mito Foundation, which funded research in Stephen Gray's lab at the University of Texas Southwestern Medical Center to found Taysha gene therapies to accelerate research into these conditions. But with the relative crash of biotechs, Taysha had to make the hard decision to lay off staffers and actually then really restrict the number of rare diseases that they could look at any one time. So rare diseases have been really buffeted by market conditions as well. So this is a really tough area, and obviously, I mean, kudos to the families who do this, and best of luck. But that's all I really wanted to say on this.
Well, I think you make a good point about any sort of economical downturns. These are the type of patient populations that are probably affected the most or are affected the most. And these types of foundations hopefully will keep championing that even through all of this. I hope that's the case. And, there was one other, the Benson Foundation here, John, but it's okay. That's a French company, and there's a really cool line. The only reason why I bring it up is because they've raised a bunch of money and they say we're not reliant on bake sales, which I thought was really funny by one of their founders. But John, interesting stuff. Rare diseases are near and dear to our heart. We're very fortunate to help a number of, I don't know, we probably have over 10 different drugs that we're helping in the rare disease space. So it's some of the best altruistic work that we can do. They're not big trials, John, by any stretch, and they're really frequently very tough bioanalytically, right? There's for a variety of reasons. Sometimes they're in pediatrics, so you got sample volume issues. You got to rely heavily on preclinical work because if there's only 1,000 people in the world that have this disease, your phase 3 clinical trial, you can't do, you can't be a double-blinded placebo type trial, right? That's just not feasible. They usually
or ethical in many cases.
Yeah, exactly. Well, yeah, very true. It would be incredibly unethical to help one and give someone a placebo. That's a good point too, John. But phase 1 trials are small numbers, and then they go they do like a phase 1, 2B, and then it goes right into 3. And phase 3 trials might have 10 patients or something.
Yeah,
and oftentimes that takes several years to even accrue those patients. So
yes,
really, really. I think it's noble work is what comes to mind. So good stuff.
Yeah. So on the when you said complex bioanalytical, that really takes us on to our main topic, which is a resumption of where we left off last time, and the evolution of drug design and its impact on bioanalysis. So we touched on a number of things last time, talking about small molecule drug development, the analytical tool development, and small molecule bioanalysis transformation, the emergence of large molecule therapeutics, brief history of drug approvals, and then drug design to improve PK characteristics. But one thing, and just to kick things off, one thing that has really changed bioanalysis over the last. I'm trying to think the first time we worked on anything. It'd probably be since around 2005 for us as small molecule people, is really the emergence of the importance of biomarkers and then the bioanalysis of biomarkers to really not just show where we started doing PK studies and showing what the drug was doing, what the fate of the administered drug was in the body. When you move to biomarkers, now you're looking at the effect of the drug on other endogenous molecules in the body, and is your drug affecting the body the way you think it is? And so that's where really a lot of our work now comes from. It's, I don't know what the split is between PK and PD, but it's where in 2005 where it was dipping our toe in the water for the first time. We're way beyond the single study.
Yeah.
It's a lot of what we do.
So, John, I think we on the biopharma side, it's probably it fluctuates between a 60-40 or 70-30, meaning 60% PK and ADA. Remember, we got that ADA.
Sure, yeah, absolutely.
And then it kind of fluctuates month to month, but we're still the lion's share. We're almost always more than 50%. I can't think of many months where we were over that 50% mark on the pharmacodynamic and biomarker space. And then I think on on the pharma side, it's probably fluctuates between 20 and 30, right? Or maybe even 10.
I think so, yeah. It's not a high proportion.
Amount. Yeah, and then I think Don's teams, the hybrid mass spec is probably a split right down the middle. I bet you. I'm sure. I got a feeling about half of her work is done in this PD space as well.
Yes.
But before the term biomarker was ever really even coined, back in 1994, when I started at a cell and gene therapy company, we didn't have PK. It was all pharmacodynamics. So I'm almost, you mentioned 2005. Actually, I kind of started, and that's probably why I have such a passion for biomarkers and PD, right? That was what I was doing: flow cytometry. I was doing proliferation assays, all things that were pharmacodynamic markers, looking at the cells we were putting back into people along with the monoclonal antibody we were using. Mm-hmm. So this has become, for me, it's, kind of at the forefront, but you're right, John, for years, at least it's only been in the last 20 years that we started translational medicine departments started popping up. And then in the last, I'd say 5 to 7, it's become, almost a requirement, or it really, there's some staggering numbers around, the number of drugs that advance from phase 1 to 2 and 2 to 3 and 3 to approval. If you don't have a pharmacodynamic marker, your likelihood of success drops almost by a half. I think it's 30 to 40% it drops. So having this is paramount. And then there's a number of companies, one in particular, a very large pharma company, they have a pharmacodynamic marker with everything. So all their biologics have PK, ADA, and a PD assay. It's just they don't produce anything without that. And that's a strong way to do it, John. It's a little bit more of an investment, but that's absolutely the way to do it. And just I'll kind of give it back to you in a second here, but why, you kind of touched on it, but just looking at pharmacokinetics is great. Even looking at is there antibody being produced on the biologic side, small molecule, okay, do we get a good PK profile? And is it lasting? Does it meet a kinetic we like? But ultimately, what's the measurement? Is it impacting the mechanism of action? We talk about it as the biology sometimes, John, right? But is it augmenting the patient in a beneficial way? That's how I think of any biomarker PD assay. It's they have you can measure the patient with a certain level of the marker, and then either it goes up or down based upon what your drug's mechanism of action is. And what a powerful way to show the efficacy of the drug. I know that's simple, but I hope the audience understands that, you hear this term biomarker all the time. Maybe you hear pharmacodynamic, but I think there's a slight difference. A biomarker in general could be a whole host of things that they're still trying to kind of discover or exploratory biomarker or research biomarker, right? That's kind of one bucket. And that's where you might be looking at, say, an immuno-oncology patient who's got high levels of chronic cytokines and low levels of acute, because that's what happens. This chronic inflammation turns into tumorous, right? And so they're trying to kind of flip that. But a pharmacodynamic marker is a little bit more, let's say, critical. And that's where it's if I'm trying to augment a T cell or someone's immune system to produce something new. And in a small molecule setting, that has a lot of, how do I say this? It has a lot of impact on phosphorylation and things like that in your world, John. So there's a lot of different sort of mechanisms of action, as well as pharmacodynamic markers that are fairly critical to the efficacy. Well, not just fairly, they are critical to the efficacy of the drug. But oftentimes, it's only within the last few years that I feel like groups are investing more in it, because they said, "Hey, look, in vitro wise, I see it." And then also, we're going to touch on this in a minute. It was hard to get samples, John, right? And
Sure.
that was part of it too. It was like, well, we think it's working, but how are we going to get these? And now that we've moved more into, we're going to touch on this, non-liquid matrices, as well as even just a further advancement in our ability to look at whole cells and things like that. So I'll take a small breath, and all of this is incredibly important to the evolution of drug design, and how biomarkers, translational medicine, and pharmacodynamics have now become, paramount to the success of drugs, to the point where most companies have a head of translational medicine.
Sure, and when we talk about effect on bioanalysis, obviously LCMS for, at least to begin with, was a smaller piece. We're talking small molecule uh, biomarkers, and it was interesting how those of us who knew about like spiking into solution extracting, standards, QCs, and running for PK assay. Your immediate thought was doing the same approach for these endogenous compounds. So it was like, but you've got this baseline, so you're adding on top of a baseline with your standards, and that we had a couple of assays like that, which was it's unsatisfactory, because from patient to patient or from lot to lot of matrix, that baseline changed. So you're having to do like downstream mathematical calculations to compensate for that endogenous amount that was actually in the matrix you were working with. So there was two paths to early on, was looking at stable labeled versions of analytes to act as surrogates for the endogenous, or the other approach was to use a surrogate matrix, where you're looking at you're preparing your calibration standards in something other than, say your samples are in plasma, you're preparing them in another solution, be it like BSA, PBS
Yeah.
mix, and then so because you can guarantee in that BSA PBS, your standards are starting from zero. Your standard one has no background that you're relating to, and then you have to show parallelism between the surrogate matrix and the matrix you're measuring samples in, and then you have a path forward. So that it did change people's mindset as how they approach these these kind of situations, and it's again made you had that little hurdle, how do we address this, and then it became a much more routine thing. Then it became an educating the customers, here's how we would approach endogenous molecules, it is more complicated and here's the reasons why.
Yeah.
It's not just when it comes to a PK assay, you're adding an exogenous compound to a matrix. It's a much simpler challenge than dealing with something that is already in that matrix, and then how do you get around that? I do want to keep sort of coming back to how it affects our approach to bioanalysis wherever we talk about any of these things, but that's where the biomarker piece adds the complication for us as scientists.
Yeah, and John, I think you're touching on some really important stuff. The industry, only within the last 4 or 5 years, has come to grips with a pharmacodynamic method will not need the same bioanalytical performance characteristics as a PK assay. And
Absolutely.
we still encounter periodically people who want to do that, and it's unnecessary. As you mentioned, there's almost always some endogenous levels. When you do look at PK, the matrix is very clean typically, so there's a huge challenge there. Overall, you talked about using, like, I think of it as like a surrogate or a heterologous curve, because you have to use a non-matrix, and that's one way to come. Or you can look at endogenous levels and do some baseline subtractions is another way to do it. And so this is something that the regulatory landscape of this, if you don't truly understand it, I think you can inefficiently develop and validate your biomarkers. So I think people sometimes do way too much work or not enough work and everything in between. But there is certainly, it's an evolving field, and we could probably, maybe we should have a podcast just on, the evolution of biomarker itself in terms of where we were, say, 10 years ago when the first guidances or white papers started coming out to where we are today, because dilution linearity doesn't mean anything for a biomarker, John, right? Because it's all about parallelism, like you said, in the endogenous level. So that's one really important differentiator. The other is when you think about the endogenous levels, there's many ways to handle it regulatory wise. The first is we take what we prefer is to have, if you can procure matrix from either healthy or disease that have high, mid, and low levels of the endogenous, because that's not, I'd say like maybe a quarter to a half. I don't know what the number is, but not all biomarkers have that.
Sure.
But a good portion of will have some level, but it's all either low or really high. So you got to dilute it if it's really high. And then if it's really low, you're kind of stuck. You might be able to, we do some things where we stimulate cells, or we might try to proliferate them in order to get the endogenous level up in vitro. That's one way. Another, that's if you're going to do that, that should be a method that's for inclusion, exclusion, and primary endpoints. That's kind of the type of method you'd need there, where that's a very big road to hoe, so to speak, or a long road to take to generate in vitro stimulated whole blood or PBMCs or a cell line in order to generate the endogenous levels. That's an investment. But on the other side, the other thing you do is like we want to take a hybrid approach. You got to do something endogenous wise. If it's not present, you can screen a bunch of normals and show, hey, we're gonna maybe handle it in the disease setting. But when I say a hybrid approach, it's a combination of an endogenous QC and still having the traditional matrix spiked QC.
Yeah.
What we like to do is a high and a low of the endogenous and a mid spiked, and a mid matrix QC or a plate QC, however you want to call it. And the why behind that is, John, invariably over time in my world, as you go from lot to lot of stuff, you might be able to, especially on kits, a lot of these are vendor supplied kits. You might have to do the trial for, say, a year or two. It's 6 months shelf life. So you've got to go lot 1, lot 2, lot 3. Usually lot 1 and 2 is no problem. But by the time you get out to lot 3 and 4, maybe a reagent's no longer available. They made some sort of change. Invariably, there's changes to it. And sometimes the endogenous QC will fall out. And if you don't have a matrix QC, because the kit still passes the vendor specs because they've adjusted the kit. But in your bioanalytical method that you've been running for 6 months, a year or whatever, it's going to fall out. Your endogenous QCs are not going to recover at the acceptance criteria that you set. Typically, some people do it as an ISR, like 67% or whatnot, right? So then if you don't have the plate QC in there, you really can't verify how you're doing. And then Dom, what do you do? Then you have to adjust your endogenous QC, and that's like real CLIA practices. This is really hard for John, right? You don't like doing correction factors in your world. That's a no-no. As a small molecule guy, you're like, Dom, but as a large molecule guy, I'm like, hey, that's been my practice for 25 years now, or 28. Like, it's completely okay to take a number and just be like, okay, but what are you going to do? I can't go back and make the capture antibody. I can't go back and get that reagent, right? So maybe I'm giving a little verbal diarrhea, but I hope that made sense, John, because there's a lot of complexity when it comes to a biomarker.
Yeah, I know. I want to move on to the next piece, but I do want to say a couple of more things about the biomarker, because again, this is the challenge for us. So for some biomarkers, you see the change from disease to post administration of therapy can be a significant change. So it's relatively easy to develop an assay for that, because the change between diseased and where the drug is having an effect is large. But in other cases, it might actually be a small change, and it's a question of, can we actually develop an assay that with sufficient rigor that measures that small change that really answers the question for your customer. That can be tough.
As obviously we could spend the whole time talking about biomarkers, but I think we got to move on. Why don't we talk about non-routine matrices?
Sure, that's fine. So yeah, again, it's having been this field for a long time. It's the requests from clients evolve, and that you see where the focus from across the drug industry changes. So early days it was all plasma and very, very, very occasionally urine. Urine became a little bit more requested with time, but then as people are more focused on, is the drug reaching its target in terms of tissue or anything else, then we start getting into the realm of much more challenging matrices. And so tissues become a big deal. We talked about fecal matter earlier on. I mean, our first request to look at feces as a matrix, certainly for me, my career, thankfully I was out of the lab by this time, was probably only about 6-7 years ago. It's the we there's so many customers you talk to now. Tissue is a big part of the analytical needs.
Yeah.
So that again requires more rigor in terms of how more challenge in terms of how you approach the bioanalytical, not necessarily more rigor in terms of acceptance criteria and things like that. So you're not necessarily going to validate, but it is another challenge in terms of what does it mean to spike into tissues, and standards and QCs and all this, and how relevant, how real are your data from that. I'll pass it back to you.
Yeah, no, and I think there's been advancements in taking tissues from humans, right, non-invasively. That's really what spurred a lot of this. We just talked about how people are inserting paper with liquid nitrogen into your nerves, John. So we are now able to kind of take out tissues. More and more tissue samples are being taken without hurting the individual. And to your point, the qualification, because it's rare that you will validate a tissue biomarker because of its innate nature, meaning you just touched on it. It's not, when you go to you got to homogenize it, right, John? So It's not like I could take it. There's no value in taking the tissue, spiking in the calibrator or standard into that, and then homogenizing it. That's not what, what you do is after you homogenize it, then you spike it in. But, our goal, if you think about it, working backwards, one of the keys to success in bioanalytical is to work backwards, right? So you've got to try and mimic how a true patient sample is going to be taken and then measured. And you got to try and create standards and QCs or calibrators using the same mindset. And so when you got a tissue biopsy, there are several challenges. It's not like I can line up a bunch of normal donors and collect whole blood or urine or whatever. I can't go in and grab a bunch of tissue. So that becomes the first challenge, right? And then the other piece is just what you said. How am I? It's not like I can really create a true standard curve of any sort other than creating a bulk of tissue. There is ways to do. You create matrix, and typically that's just for QCs, John. You just verify that you do some development work to say, hey, my standards behave similarly. I got to design the assay because I don't have enough matrix. So if you can get away, the goal is to design the standards in a buffer of some sort that's the equivalent of a matrix curve. And then you need matrix QCs. And then, in preclinical studies, we can talk about soft tissue, hard tissue, and all that sort of stuff. So great stuff, John. I hope that made sense.
Yes. I don't think there's much more to. I mean, basically for us, I think the key is, as bioanalytical people, we want things to be liquid, and tissues aren't liquid. So the challenge is, what do you do to make liquid that is then relevant?
Well, then there's a whole art form in getting the tissue into a liquid, which we're very good at KCAS. So that's the other piece, But I think we've touched on that. It'd be good to maybe move on to some of the targeted therapy.
Absolutely. Let's go ahead. And again, drug evolution. It's what we talk about. We talked about measuring tissue matrices because people are interested in has your drug reached its target. There's a lot of emphasis on developing drugs that are targeted to get to a particular place, with antibody drug conjugates being a prime example of that. I'll pass it back to you.
Yeah, I mean, the ADCs are revolutionizing medicine, along with even just advancements in monoclonal antibodies. And I believe in small molecules, John. They've gotten a lot better about their potency and even like half-lives. And so, but targeted therapeutics aren't just limited to antibody drug conjugates. There are other things. You could take small molecules and put a linker onto that and then add something to stabilize it. That technically falls into more of a targeted thing. There are people taking pieces of the antibody, like an FC region, and linking, the FABs, and linking them together to form like a multivalent. and then popping an ADC on, some sort of small molecule onto it. People are taking antibody backbones or the framework. That's what the antibody is in the ADC, that framework. You don't just have to put a small molecule payload. They're putting, radio label, some isomers on there. They're putting all sorts of stuff on there to help just deliver the drug to the area of need rather than systemically. Even though you might give it systemically, there's a much more benefit of having a ride to right. That's what this is. I think of it as it's a ride to bringing the payload to its destination. And then I think we're getting into sounds. We talked about that a little bit, where there's a lot of like pulverization with lasers that can be done, stimulating your mitochondria. I love that one. So all cool stuff when it comes to, maybe there are even things where you can systemically give someone and then shine a light on the tumor, and that'll help kill it. I mean, great. It's just, those are more dermal at this stage, but you could think of like, there are people as we sit here are trying to think about how can I cure someone's lung cancer with this, right? So this is all really amazing stuff, John. So targeted therapeutics, they're not going away. It's a common, what better space to be in than when taking all your large molecules and small molecules and combining them, right? So it's just common sense for me now, but those are fascinating things. And John, last but not least, you can jump into cell and gene therapy. So
just the last word on the like that targeted therapies, and the fact that these you touched on so many different examples where these combinations of various pieces. For us, the analytical challenges quite often being asked we have to measure those various pieces, and then that becomes the use of various different technologies to apply to measure those pieces for example, to touch on ADCs briefly, that purpose is to develop a very potent, and we've touched on in the past, very potent toxic payload to a site of interest. But what you don't want to do is have that toxic payload drop off before it gets to the site of interest. So actually the measurement of that free payload is absolutely a key part of the development to ensure that the linkers and everything that has been employed are correct, to ensure that payload gets to the target, and there isn't instability within the wider molecule that it falls off beforehand, because that has severe implications for patients.
Yeah, John, just like anything that's great, or ADCs are great, but bioanalytically, they're like 4 times as many assays, right? You've got a free and a total payload on the LCMS side, and then you've got to measure frequently there's a total antibody and then a total ADC measurement.
Yep.
The naked antibody as well as the antibody with the small molecule on it. And then there's the immunogenicity. And the immunogenicity is typically just a standard ADA against the framework of the protein. But if you get successful, then your ADA and your immunogenicity profile gets much denser. You got to start looking at the linker and domain specificity. So even as the drug becomes successful, the bioanalytical pathway gets larger, unfortunately. Yes. So ADCs, despite how powerful they are, they also have that, like as you mentioned, they have a little bit more risk to them, right? Innately, they have more because the payload can fall off. And also, if the biologic's binding, that has... better make sure that biologic's find in the right place. So all sorts of slightly more nuances, but John, the reward is palpable, as you know.
Absolutely. So I didn't mean to deflect you from.
No, that's good. I think we can cover gene therapy real fast here.
But now you can have free reign and off you go.
Yeah, no. So cell and gene therapies, near and dear. Although I love monoclonals and ADCs, it's been what I've been doing for a long time. And so the challenge here is there is no true PK assay, right? Yeah. And that you can't go have someone make an internal standard, or even in our case, you can't make for instance an anti-id to capture a protein. That's not going to work. Or there is no way of, if the therapy itself is some genomic material, and we can talk further about what I mean there. And if it's just genomic material itself, if I put 1,000 copies of that in, there's no way I can measure. I'd be lucky to measure any of it as it goes into an individual. So that even measuring residual amounts, but you're not going to get a PK profile, John. Let's be very clear about that. Yeah. And then when you put cells in, it's the same thing. You'd be lucky to even measure those cells. And even if I can measure them, if I put 50 million cells into somebody, I'd be lucky to measure 1 or 2 or 10 or whatever, but it's not going to be any sort of PK profile. And certainly within a matter of days, it's all going to be gone anyway. So what do? Right? The key here is that pharmacodynamic piece. So that's when you start looking at, you really got to start, to me, I think, and then I've been reading regulations on this, John. I've been reading a lot of the regs. And the first statement is the regs that says, for cell and gene therapies, it should be taken on a case-by-case basis. And when I read that, I went, oh, no wonder. I think, John, I just innately do that. And I know that sounds crazy, but when I talk to our clients, I'm not thinking about regs. I'm just going. I'm just like, oh, tell me more. So when you do these things, the way I see it is, you got a patient or a healthy, and you gotta characterize that. I look at it as their immune cells, because I'm biased here, or some sort of soluble biomarker. But you've got to get the patient, and it can't just be one. It's got to be, to me, somewhat multifaceted. That's why I like flow cytometry so much. So even if it's like some sort of T cell, B cell, NK panel, along with other subsets of cells. And you can get specific T cells. You can look at T cell receptors. You can use things like tetramers to see how many specific you might have. But you're trying to augment that person's, typically in immuno-oncology specifically, maybe I'll caveat it there, you're trying to augment that person's immune system to make de novo cells to combat the tumor. Mm-hmm. Or you're trying to knock out cells that are bad, because that's another, it's a little bit of both. Or you're trying to ablate certain cell populations that are overriding others. Those are hematological disorders. But when you do those types of things, the key is to measure it based off of the baseline sample and then the experimentals. But all along the way, you've got to develop a sophisticated approach, not just on that cellular piece, but I do think you need to have some other critical soluble biomarkers, or even some of that non-liquid matrix biomarkers, where you can try to get a piece of the tumor and look at it pre and post, or for example, maybe a lymph node, or these are things. But I know if I'm sitting in a hospital bed and it's life or death, you can take pieces of me, John. So those are some. I hope I kind of started to paint a little bit of a picture there, but to continue with the thought, it's very when you go to look at patient to patient, John, maybe I have, let's just say 50 circulating anti-tumor cells. Say we got a method that can measure. Do I have cells that are against whatever I'm trying to treat, right? And then maybe you have, 500. And maybe I vaccinate you and you get 50,000, and I vaccinate me with my existing 50 and I go to 5,000. I may have more protection than you, John. That's where it's tricky.
Yeah, sure.
Because and for the longest time, especially when I was in grad school back in the late 90s, early 2000s, you could easily vaccinate someone with a peptide or a protein and generate a circulating immune response that was not protective at all. In fact, it wasn't. What they started to do is find ways to overcome some of the microimmune, like the tumor itself, it wasn't getting there, and that's why it wasn't protective. And then we got more creative than we started, even if you pulsed a patient's cells, right? You could take out someone's cells, generate, and this is like what CAR T cells are they're not quite there yet, but you could put in, cells in vitro that look like they're going to kill the tumor, and they still couldn't. If you tried to inject those cells into somebody, they still didn't work. It's not until the CAR T cells came around that you were starting to get cells into people that were integrating into their immune system and getting true circulating, I think central memory is what that augments, right? So some of those other things are just transient responses that can overcome whatever the tumor is doing, the micro tumor environment that's hostile, John. So even if a cell could get there, it's getting destroyed. It's that systemic memory response that allows for it to mount the proper response. And I don't know, I just probably summed like 20 years of immunology. I don't know how well I did, but I hope that made sense in terms of what it kind of looks like. Like pharmacodynamics are tough. And John, a lot of it's done preclinical, right? So a lot more work is done preclinical if you take the right approach. So getting back to what clients should be doing, you can do a lot more preclinically and then try to get some of it to translate into the clinic. Sure. And clinically, it is that you got to take that approach of, if you set an expectation that I think every patient's going to get 50,000 circulating specific tumor cells, you're going to set yourself up for failure. So.
Yeah, absolutely. Yep.
So I hope that I'll stop, and by no stretch, I would love to debate this more. That's just my opinion. It's not KCAS. There's many different philosophies and many things that you could argue for and against that I just said, because this is a vast field.
Yes.
I'm sucking the air out of the room, John. I can feel it. I'm sucking the air right out of the room. You're like, Dom, I don't know what to tell
you. but again, you're right. Is the pharmacodynamic piece is key as well, and so that's where a lot of the opportunity comes for us with the more conventional technologies, is because we're looking for again the effect of these therapies and markers.
John, I should caveat it by saying there is some, now that we're using more AAVs and certain vectors, you can do PK on those. But again, it's not going to be a traditional PK because you're just looking for copy numbers to be present or absent. And that's part of that biodistribution, off target, on target. Some of it's more safety issues, right? That's part of what some of the pharmacodynamic pieces, and they get commingled. You really should, a lot of your pharmacodynamic assays are in part safety methods as well. Because, there's no anti-drug antibody responses in cell and gene therapies, but immunogenicity is there's a lot you need to do immunogenicity wise, especially if you're treating rare diseases, which we know about 20% of all cell and gene therapies are in this space, right? Mm-hmm. If you're trying to knock a gene in, that's an immunogenicity risk, because if someone's absent, a lot of the rare diseases, they're missing an enzyme or something like that. You knock that enzyme in, that person now sees that has the potential. In fact, almost all of the patients will generate an antibody. Is it neutralizing and so forth? That becomes risky. And then back to the pharmacodynamic, there are other platforms other than flow cytometry. ELISpots are wonderful things. And then I talked about the PCR is another platform, but there's more and more emerging. I think some of it I'm going to stop because it's somewhat proprietary, but people are getting more creative. But as we stand today, there are two true and tried methods of measuring, patient-specific augmentation of T cells, de novo production of T cells, and that's flow and ELISpots. Those are still the tried and true method to do it, and we offer both of them here at KCAS. Shameless plug.
There you go. You got to take advantage. You have the air, so you can say what you want.
No, John, this has been really a lot of fun. I don't know how much if we want to wrap up, or I know you. Let's wrap up. I want you to tell me about your exciting weekend. I haven't had a chance to hear about your rock star encounter. Why don't you tell us a little bit about that?
I think it is moved to wrap up. I don't think there's much more to say on this, but when we started on this topic, I didn't know it would take 2 weeks, but it has actually been really, really enjoyable and great to cover. I don't know if there's anything else you want to add to that. I mean, it's amazing. I will say my piece, actually. It's amazing I've been in this business. I started doing mass spectrometry in 1985, and the advances in the instrumentation in that intervening 35 years has been astonishing. So it's going to be, I can't wait to see where the industry goes and where Bioanalytical goes with it, because the things we can do now are just incredible compared to what we could do when I started out.
I imagine a 7500 is like complete Star Wars like technology for you, right?
Well, yeah, I mean, well, you think about just to touch on it very briefly, it's the triple quadruple didn't exist, I don't think. Again, you think about just technology in general. No, our phones, they're more powerful than the computers then. And I remember the disks that we used to store mass spec data were probably about 15-inch diameter, and they stored kilobytes, and the actual drive for it was 5 foot tall. And so that's a piece of it. The fact that we can now routinely interface liquid chromatography with mass spectrometry is huge. I mean, that was when I started doing mass spectrometry. That was the key focus. How can we make this work? We know we have these two technologies that, if we could marry them, would be fantastic, but they're on paper incompatible. So that the emergence of the electrospray and ion spray sources were transformational, and obviously instruments have moved beyond that, and then that moving from small molecule to large molecule. everything else. So I'm not going to say any more because otherwise,
No, I think it's a whole other subject. But you got me thinking about the first. Yeah,
LCMS is involved. But that's the excitement of this business. It's not just the revolution in drugs and how they've evolved, and design of them is involved, and medical treatments is involved, but how our technologies that we work with on a daily basis, how they've evolved to meet that need, so we can routinely measure these things.
yeah, John, you really got me thinking about the first flow cytometer that I worked on was a 4-color. It needed a dedicated room with low lights and lots of cool. Because of the size of it, it was huge. It's probably the footprint of, and it did 4 colors, and it took. There was some. This was '94, but the computer was massive. But we, floppy disks and everything else. And that even the evolution of that is funny. But the system itself was like this was it was called a Coulter Epix XL, and it had it was an elite, an Epix, the elite system, and it was a Palm Pilot that you used for gating. Essentially, it had like a pen, the old pens you used on Palm Pilots, and it had a small screen that you would touch on and circle the cell populations. Well, yesterday I had a call looking into automating that gating for something that we're thinking about investing in, and just the thought of how far that has come. We're up to like 60 colors, back from 4 up to 60, and the amount of data that we're generating and what we're looking at, it just makes my head hurt exponentially in terms of the sheer volume of data bits that we can generate from we're at 18 color, but a 60 color panel, there's no way you can do that manually, John. It has to be done.
Sure.
Some sort of artificial intelligence. So, all right, John, tell us about, this was great, man. This was so enjoyable the last few weeks. Tell us about what you've been doing.
So, as we talked about last time, the big event for us was Mark Burgess of the Chameleons coming and playing locally. So a very quick background to the story. He and his band were planning to tour in the autumn. He actually is living in the US because his partner's over here. And so he said, I would like to do a fundraising tour so I can actually help pay for travel, visas, transport, all the stuff that you need in a full band tour. And he said, contact me if you're interested in me playing your backyard or any venue. I'll play it. So I contacted him, said totally interested. Then it went quiet, and I think I contacted him a couple of weeks later, and he said, are you still interested? Are you serious about this? I'm like, absolutely, I'm serious. So fast forward several weeks, July 22nd was the day we were given. For actually a couple of other complicating things, I decided not to do it in our backyard, even though that would have been very nice. We actually got in touch with the local venue, which has been legendary for years, although it's now a brew pub called Garrett's. So we talked to them. When we look back at it, it's really funny. I just walked in one day to Garrett's and said, would you like to have a band play this Friday night? And Greg Garrett, the owner, says, yeah, that'd be fine. And that was as much of an agreement as we had between us. So when it came to the Friday night, we had a guy contact me out the blue asking to be a support act, a guy called Alex Reed, who's a professor at Ithaca College. So he was the support act. He set up the sound and everything ready for Mark Burgess and Stephen Rice, who is his bandmate, who are actually, while we were setting up, were driving over from Boston, because that was where they played the night before. So that's a 6-hour drive. So it was like one of those things that will they turn up in time? So there was always that hanging about. Anyway, they turned up, or I think a little bit after 5 o'clock, stopped off at the house to drop off baggage and stuff. So they met all the family while I was at the venue. And then they turned up at the venue. We did set up, did the sound check, which was, honestly, he did a cover of David Bowie's Ashes to Ashes, did a couple of his own songs, and the sound was immaculate. And I'm thinking, this is going to be great. And it really was. At all levels, it exceeded expectations. I think we didn't have a huge crowd, but I actually don't think you wanted a huge crowd. It was nice, relaxed atmosphere. They came on stage, a little raised platform. They came on the little raised platform around 7:30 and played to just shy of 10 o'clock. They had a 50-minute break in the middle of it. It was Mark Burgess and I'd like to say his bandmate Stephen Rice. really nice approach to, you know, if you're a fan of the band, they released 3 albums in the 80s and a few more things since then, but he was giving background to some of the songs, so it was like you had this it was just great to hear, here's what the song's about, etc. So again, it helped to the relaxed mode throughout, and just it was a really, really special concert. I have to, it's possibly the best concert I've seen, just because of the feel of it, you know. And Mark Burgess is a really down to earth, friendly person. Stephen was really nice too. So you had these two people who were obviously really enjoying themselves. You know, the concert finished and they were selling memorabilia and chatting to fans and stuff. We then ended up loading the van. We came back to the house, and then, because the whole thing, Mark couldn't eat before he could play, so we'd arranged with the pizzeria next door to, I actually went into and said, what time are you open to? And they said, oh, well, last order's 8:30. I'm like, fine, we'll be there before 8:30. So we got my wife, I'd arranged with my wife, she'd go and do the order. She went to like 8:15. They shut, and it's like, you got to be kidding me, we were relying on your pizza. So then it was like, she and I talked. She quickly ran up to the restaurant called Little Venice, which is Robert Moog's workshop where the original Moog synthesizer was built. So she went there, ordered food. But while she was out, the one song that she wanted them to play, they played it while she was out at the building. So like that didn't go so well. Anyway, we brought the food back to the house, cracked open some wine, we ended up chatting, we ended up on the back step, and while we're talking, we eventually persuaded Mark to bring out his guitar, and he played some songs on the back step of our house. So we had like this little secondary concert of about four songs or so. And then I have another story which I'm not going to share beyond this team. Once we finish this podcast, then I'll tell you about that.
No, John, I got to tell you, that story is amazing. You're like that's like everything that's great about the world, not just America. Like that's sort of it reminds me of maybe what like the 60s were like, when people were just free loving, spirited, and all over the place. It's refreshing to know that you just welcomed kind of like a stranger, albeit a celebrity, and just had you could hear it in your voice how you're like, it was the best show I've ever been to. John, that's just congrats. I'm glad it went, despite the hiccup with the food. It sounds like it went great, and I can see you hanging out on your back step while there's like, I don't know, it'd be the equivalent of maybe a member of like Whitesnake or Iron Maiden or something like that playing for me. But like that's unbelievable.
So just a couple of post notes. So they started in Texas, I don't know, 3 or 4 weeks ago, and it's just two of them been driving. And Mark was saying, from his perspective, this has been the most fun tour to do, because he's seen the country while he's doing it. You know, with the first time he toured here with the Chameleons, they just did it all by plane, so they didn't really see much at all. So he's really loved that aspect, and I think that helped to the whole relaxed demeanor of stuff. My eldest son said that for him, he's got like little certain things have happened that he'll carry with him for the rest of his life. Going to Madagascar, which I've mentioned numerous times, that was one for him. And he said this whole Mark Burgess concert and just the whole piece of it, that's one as well. It is a once in a lifetime thing, and it really was fantastic.
And I love the small venues, John. You're right. I think back when I was living in Boston, I got to see like Eddie Vedder play in front of like 800 people. It was amazing. Sure. I'm sure that's still very small for what he was at the time. And those are always some memorable moments. But John, we're running long. I'll just share. I won't share much, other than I think I don't know if I mentioned it. I picked up 80 pounds of chicken wings while I was in Nebraska over the 4th of July, and so they were $2 a pound. They're $6.50 in the grocery store here. I have been making chicken wings. I like the butcher I bought them from told me to thaw them to a slurry, and then you could refreeze them. So I have like 15 or 16 packs of like 2 to 3 pounds of wings that are all frozen and shrink wrapped and then safely in my freezer. I'm not going to starve. But man, I've been making buffalo wings that my wife and son, I got my buffalo wings badge is what my son.
We love wings.
I even sous vide some recently. So I've been smoking them, and I got a whole process that I used. But last I used them in the sous vide, and I always like finishing stuff in the oven. It just gives it I put the sauce in there and make a little tinfoil kind of packet, a hobo tinfoil, and just then it's ready to eat whenever you're ready. So that's been pretty much where. And then I turned 50 between now and the next podcast. So although I don't know what I'll be doing, but the wife is kind of planning some of that. But then end of September, the Warino men that are over 21 are going to meet in Vegas. So to be determined there. Yeah, that's my little. Yeah. I might miss WRIB because of it, but such is life. I've been there the last 9 WRIBs. So, but I'll that's still TBD. But I don't think I'm going to make it because that coincides with it kind of overlaps with the trip. So I'd either have to where is WRIB this year? I
can't remember.
It's in San Antonio. I forget, but I'd have to travel directly from there back to Vegas, and I'm too old. John, I got to tell you, I really don't. My days of spending from city. I remember I used to take trips I would fly into New York, spend a few days there, maybe go down to Philadelphia for a few days, and then fly up to Boston for a few days and fly home, or go to San Francisco, San Diego. And I can't do that anymore.
I used to do sales trips, and I'd do a country a day.
Yeah, I can only imagine. I'm complaining about going city to city over a 5-day stretch, and you're going from country to country. But John, it's been a pleasure. It's really congrats on your wonderful visit or hosting. That sounds so incredibly memorable, and what a great couple podcasts. This was some of my funnest. This could be one of the funnest series we've done. I hope our audience has enjoyed it. Last call for you, John.
Absolutely, no, I agree with you. I think these have been great, and I hope, like you say, I hope people have enjoyed listening to it too.
All right, we'll see you in the funny pages, everyone.